Hello everyone, I once again welcome you to my series of lectures, that is Understanding Pharmaceutical Science with Dr. Hariharan. So today's topic we are going to discuss about the cell-mediated immunity. So I have divided this topic into two major segments. So first we understand what is meant by immunity and the types of immunity. Second, we will discuss about the cell-mediated immunity in detail, especially its mechanism of action. So first, immunity is simply a state of defense mechanism against the foreign substance, that is an antigen. So we can classify the immunity into two types, that is, one is the innate immunity and second is the adaptive immunity. So if you see the innate immunity, it provides a first line of defense against the foreign substances. It is non-specific in nature. Whatever the foreign substance enters into your body will be stopped by this immune mechanism, then it is eliminated from our body. It is non-specifically. Generally, it includes our skin, our mucous secretion, gastric acid secretion. Everything will come under the innate immunity. And the second type of immunity is the adaptive immunity. It is the second line of defense mechanism in our body. It is highly specific in nature. It specifically eliminates the foreign substance. It specifically eliminates by means of identifying the surface antigenic determinant site called as epitope of this antigen. And moreover, it also keeps in memory what is happened, what was the antigen previously it has encountered. So once it comes for the second time, it will eliminate within a short period of time. So if you see the adaptive immunity, which is capable of inducing a specific recognition and elimination system in our body, it eliminates all types of foreign substance, or whatever it may be, a microorganism, it may be a chemical. So generally we call this foreign substance with a single name called as antigen. So actually how it specifically eliminates is based upon a particular part of the antigen we call as epitope, which is called as antigenic determinant site. So this adaptive immunity is basically carries of four attributes in its nature. One is the antigenic specificity. So I have already told you, it specifically binds with a particular antigen. That means it recognizes the antigen by its surface protein sequence. So that protein is nothing but a polymer of amino acid. It specifically identifies the particular sequence, and against that it produces the antibody. Even a single change in amino acid of this epitope will produce a different variety of amino acid. So the second is the diversity. Since it can specifically recognize each and every antigen with a difference of single amino acid, so it can produce an antibody similarly in a large numbers. It produces a diversity of molecules of antibodies. It can produce diversity molecules of cells, which can help in this adaptive immunity. And third is it gives the memory, because the memory cells produced during this adaptive immune response helps the host, once the same antigen it encounters for the second time, it gets eliminated from the body with a short sequence of time. And fourth and the final one is the maturation process, that is the self and non-self recognition. Because mostly the 99% of the antigens are protein in nature, so our body also carries the surface protein. So what happens is that the protein which is ours is called the self protein, and the protein which is present in the microorganisms or any foreign chemicals, which is we call as a non-self protein. So our lymphocytes has to designate the difference between this self and non-self protein so that it can easily eliminate it. So these four attributes has contributed the adaptive immunity. So based on the mechanism of action, the adaptive immunity can be classified into two major types. So if you see, one is the humoral mediated immunity that we have seen in our lecture, and second is the cell mediated immunity. So we are going to discuss elaborately about this cell mediated immunity in detail. So in this lecture, first we understand what is meant by a cell mediated immunity. Second, we discuss about the mechanism of the cell mediated immunity in detail. So what is a cell mediated immunity? If you see, the virus are generally intracellular in nature, whereas the bacteria are extracellular in nature, because the virus can reproduce only inside a cell. A virus will enter a cell, then its genetic material gets copied and its covering gets copied only inside the cell, and the lysis of the cell happens, then the virus gets eliminated. So generally when whatever material which is intracellular in nature, then it's encountered by the cell mediated immunity, because since it is intracellular in nature like the virus, the antibodies which is present in the blood is not encountered. So once it doesn't encountered by antibody, so no immunity can be produced in the humor itself. So it's need a separate division of immunity called the cell mediated immunity. So we can tell that cell mediated immunity response does not involve antibodies. So then what it will do is that it rather activates the phagocytic cells, majorly the macrophages and natural killer cells, and very specifically the cytotoxic T-lymphocyte. This cytotoxic T-lymphocyte plays a major role in the cell-mediated immunity, and the stimulation of these macrophages and phagocytic cells and the cytolytic T-lymphocytes will happen based on the release of the cytokines from the antigen presenting cell. So if you see the mechanism of the cell mediated immunity, I can classify the mechanism of the cell mediated immunity broadly into two ways. So one, it involves the activated helper T cells which release cytokines, thereby it can stimulate the cytolytic T lymphocytes one way, or other phagocytic cell. That means in one mechanism it activates helper T cells. In the second mechanism, it can directly activate the cytolytic T lymphocyte, thereby it can induce the cell mediated immunity. So there is two types of mechanism is possible in the cell mediated immunity. One is the involvement of helper T cells, and second is the direct involvement of cytolytic T lymphocytes, cytotoxic T lymphocyte. I can tell it in another manner. So I have represented in the picture, as you can see, what happens is that once the antigen enters the body, in the case is an intracellular antigen, so the antigen presenting cell may be a B lymphocyte or macrophages which engulfs this antigen and it processes it. After process it present it. So it present into various type of receptors, as you can see from the figure. It can present into two types of form. One is called MHC Class I and second is called the MHC Class II. So after the antigen and processing and the presentation, it may be of major histocompatibility complex Class I or Class II. The binding of the cells, that is the helper T lymphocyte or the TC cells, will stimulate the specific mechanism of cell mediated immunity. If you see, there are two types of cells I told you. That is the T cells carries two types of surface proteins which can be differentiated by cluster of differentiation CD4 and CD8. The cells which is rich in CD4 will bind with the class 2 and the cells which is rich in CD8 will bind with the class 1. So this is why we can tell that the helper T cells can bind with class 2, whereas the cytotoxic T lymphocyte binds with the class 1. So in this figure you can see it very clearly. What happens is, after the antigen processing and presentation can happen in MHC Class I or Class II. So first we consider the Class II. So once the MHC Class II is processed and presented, the helper T cells will come and interact with it. So the inactive helper T cells, what happens is, by the interaction with this antigen, it gets activated. So once it gets activated, it releases a chemical mediators called as cytokines. So these cytokines get released and it activates both B and T lymphocyte. In B lymphocyte it goes to the humoral immunity, but in this case it goes and stimulates the cytolytic T lymphocyte, thereby the cell mediated immunity and destruction will happen. So this is how a MHC Class II will work. Whereas the MHC Class I will directly stimulate the TC cells. The inactivated TC cells gets activated by this MHC Class I presentation. Then it does the apoptosis of the infected cell. So these are the two major mechanisms that by which this process can happen, that is with the help of helper T cells and with the help of cytolytic T cells. So first we get an understanding how the mechanism of helper T cells will happen. So I have already told that once the helper T cells get interacted with the antigen presenting cell through MHC Class II, it releases cytokines. So these cytokines in particular, the gamma interferon also get released. So this gamma interferon generally produces an innate type of immunity to the nearby cells. What happens is it gets binds with the nearby receptors of the cells, and it down regulates the receptors where the antigen can bind. This one way it can do it. Second one, this also increases the ability of the macrophages to kill the infected cell. So the infected cell, that is the microbe which is ingested into the cell, gets removed by this mechanism, and it is also stimulates the natural killer cells. So this mechanism, it enhances the phagocytosis apart from the cytolytic T-lymphocyte stimulation also. So this is one mechanism where the TH cells will have a major role. And second way is the mechanism of the cytotoxic T-lymphocyte. So the cytotoxic T-lymphocytes can directly bind with the target cell, that is the antigen presenting cell, that is the MHC Class I cell. So its appropriate antigen is processed and the epitope is presented as MHC Class I, and this will get binded by the cytolytic T lymphocytes. There gets it activated. Once it activated, it can damage the cells and kill it. How it does it is that by a number of pathways. So you can kill the infected cell by numerous pathways, and these pathways basically depends upon the stimulatory signals. There are many stimulatory signals, whether it's a granular or non-granular in mechanism. So one mechanism I will explain in detail, that is mechanism involving the perforins. So if you see, perforin is a granules metal, cytoplasmic granules. What happens is that the cytotoxic T lymphocyte can kill the target cell by releasing this chemical mediator called as perforin. So this is a cytoplasmic granules present inside the TC cells in an inactivated form. Once it gets activated, it gets degranulated and it is released. Once the perforin is get released, it can trigger a sequence of reaction inside the infected cell and activate a process called the apoptosis, that is the self-death destruction of the cell. So you can see from the figure, what happens is that once the cytolytic T lymphocytes binds with the infected cell where the surface epitope is presented, so once it binds, it carries these perforin molecules, which is indicated in the red color. So what happens is that these perforins get released. So once it gets released, it gets internalized by the membrane of the infected cell. So once it gets internalized, this perforin activates a cell signaling pathway and causes the apoptosis, that is the programmed cell death of the infected cell. Thereby it can produce the cell mediated immunity. So second method is the one, this is by means of granules, and second is the non-granular mechanism it can do it. So generally the cytolytic T lymphocyte sometimes kills by apoptosis through a cell surface protein called as FAS. This FAS will help in inducing the apoptosis of the infected cell. As you can see, what happens is that the cytolytic T lymphocyte has a protein on its surface. This protein we call as FAS. It gets attached with the target cell. So in this case, the target cell is the infected cell. So once it gets attached, it sends signals. It sends signals directly to the nucleus of this target cell, thereby initiating the apoptotic process, that is the self-death process. Thereby it destroys the infected cell by inducing the suicidal nature of the cell. So this can be elaborated in the figure. As you can see, the cytolytic T lymphocyte carries the FAS on its surface. It binds with the infected cell, maybe a tumor cell also, so the MHC Class I. So this will directly sends the signal and induces the apoptosis, so the cell will die. So these are the two possible mechanisms by which the cell-mediated immunity can happen. So I thank you very much for your continuous support and the listening of my channel. So I suggest you that continuously listen. We will provide an in-depth information about the various aspects of immunology. Thank you very much.